What is up, my beautiful brainy babes, and welcome back to another episode of Sex as a Biological Variable, where we dive into both the science and the soul of being female. I'm Dr. Rosie Ray, and even after earning my PhD in biomedical science, there's still so much that I wasn't taught about my own body and brain. So that's what we're doing here, learning from the scientific literature and the lived experience of women to talk about what's really going on. So let's dive in. Hey guys! Welcome back to the show. Happy Sunday. You probably won't be seeing this on a Sunday. Maybe, I don't know. But it's Sunday today. I just went to church. The sun is out. It's a gorgeous day. How am I doing? I am on day 25 of my cycle. I'm feeling a little loody. I'm getting close to my bleed. And I feel like this one's going to be a doozy for me because last week, it wasn't that I wasn't taking good care of myself, but there was just like a lot of things, fun things going on, and I was just out of my routine. Like, it was opening day for the Tigers, and then I went to see my boyfriend, and so I was just eating a lot of things that I don't typically eat, and I know that's why my skin is breaking out, but like I feel good, so I'm excited to get through my period and be done with that. So what are we talking about today? We are talking about tracking female ovarian hormones in preclinical research models, something that I know way too much about. So yeah, it'll be super fun to talk about. A little bit of context and history for this topic. Back in the 1930s, scientific researchers knew that the fluctuations of female hormones influenced physiology, they influenced behavior, they influenced the molecular changes that are going on in the body. But by the 1950s, it was just standard practice to exclude females from preclinical research because, these female hormones, they're really kind of throwing off our data. And so scientists just didn't use females, which I get, and I've had conversations and kind of arguments with people about this, that it's like, oh, well, my biological question is something different and I'm not really concerned about the fluctuations of hormones. And it's like, I get it. Like, if you want the science for science sake, sure, cool, go use males, whatever. But if you're doing anything that has implications for human health with medications, with treatments, and you're choosing not to study females, I don't know. I think that's lazy. I think that's sloppy. And what we'll talk about is that being able to inclusively account for female ovarian hormones is really easy. It doesn't take a lot of time, and it's kind of low-hanging fruit when it comes to adding an extra level of control and another scientific question in your research. So there was another regulatory recommendation that was put in place by the Food and Drug Administration in 1977, and that recommended not using females of childbearing years in clinical studies. And so this was women from ages 12 to 50 and up. The reasoning for that was, one, because of the hormones, but then two, they didn't want to influence their ability to give birth. So that was in 1977 until the early 90s. Women were just not included in clinical trials. And so this kind of perpetuated the thought that, oh, we shouldn't be studying females. So that got repealed in 1993 by the NIH with what was called the Revitalization Act. And this required the inclusion of females in clinical studies. And so this is at the human level. But when it came to preclinical studies, researchers still weren't using females because of the messy hormones. And then it was in 2016 that the NIH implemented sex as a biological variable. Yes, the title of this show, but this policy made it mandatory that sex needed to be included as a variable, a controlled variable in scientific research. Anything that's getting funded, anything that's getting published needed to consider sex. And if you weren't using both sexes, you needed to have a really good reason why. And a lot of them were because it's, oh, that's not my biologic question, which whatever. So that happens in 2016. And what a lot of people did was okay, cool, we'll just throw females into the mix. We'll also study females. And it's that's great. for inclusivity, but if you're not accounting for these hormones that we know have such a big effect that for like 50 years people just didn't want to touch them because it was really influencing the data, if you're not controlling for that accurately, like you're just asking for bad data. For like a metaphor, it would be like if you were giving one group of your animals a drug at random times of day and pretending like that didn't have any influence on it when it's something that you can really easily control for, and then add as a layer of complexity into your research project. So it's like, I don't know, it just makes sense to me. So throughout my PhD, I did a ton of estrous cycle tracking because my project originally started asking sex differences. I learned how big of an influence female hormones had on the physiology that I was looking at. And so I'm like, okay, I guess I really need to be tracking the cycle and then looking at the different phases. And then part of my project was also interested in the effect of menopause or the loss of these ovarian hormones. And so I spent so much time tracking my girls, my ladies. I had like 40 animals, 40 females at one point where I knew where they were at in their cycle. And by knowing where they were at in their cycle, I could say like, okay, number 46, her estrogen is going to be high on Tuesday. So that's when I'm going to use her for my experiment. And like just having that level of detail, especially if you're doing a sex specific study, is like, it's so nice. It's really nice. So the topic for today's episode really came out of a conversation that I had with one of my friends, Dr. Kelly Nett. She is a behavioral neuroscientist and she's a newly minted PI, and she's setting up her lab. She's starting her research and she wanted to include looking at sex differences, but inclusively controlling for female hormones. And so we had a conversation last summer at a wedding, and this was like when I just finished my PhD. So I'm teaming with all of this information on hormones, and I'm just like spewing it to anyone that would listen, kind of like I'm doing right now. But talking about all of the influences that hormones have on neuroscience, and Kelly was like, wow, like that's really interesting. So now that she's setting up her lab, she hit me up and she's like, girl, you spent so much time doing this, like what tricks of the trade do you have? And I said, I have way too many. Let me just give them to you. But instead of just emailing the answers back to her, I wanted to have a Zoom call, record it, and then be able to put it out with a little PDF guide, which is going to be attached with this. It's also going to be on my link tree with all of the information, because like just starting to track for the estrous cycle in rats, like there's some really good papers out there, but like starting from 1930 in the 50s, like trying to go through those and like figure out what works for you is not as helpful as just talking to someone that did it for years. And like I was lucky enough to have people that I was able to learn from and talk to about it. And so I just wanted to put everything together. So if any preclinical researchers are trying to incorporate the estrous cycle into your studies, hopefully this can be like a beginner's guide for it and help you. So yeah, that's that. I hope that you enjoy this conversation. It does get extremely into the weeds, but like that's where we want to be with this information. So yeah. Let's go.
Yeah, you look good, girl. Are you in your office?
I am, yeah. Like the whole I got a nice little view
Yeah.
there.
Oh my
Got like
gosh.
stuff there to make me look like a scientist for real.
Yeah,
Alexa gave
yeah.
me these.
Cute. Uh-huh.
Yeah, yeah. So
Very, very professional. Okay, so remind me, what institution are you at right now?
yeah. So my place is I'm at Gustavus Adolphus College.
Hmm.
So small liberal arts only, well, I guess primarily undergraduate. It used to be only undergraduate. They just
Hmm.
started a athletic training master's program. So,
Oh, cool.
but otherwise just primarily undergraduate. This is where I went to school also.
Awesome. Cool.
Yeah, yeah.
That's awesome to be able to go back and I guess kind of foster students. And so now you did a postdoc out here in California, yeah?
Yes, at UCLA with
Hmm.
Dr. Kate Wassam. And so grad school, right? Like working with Alexa, I worked on
Hmm.
the addiction pathways, like inhibitory control and all of that. And then at UCLA, took a step further back into basic science, right? So
Hmm.
eliminating the drug self-administration aspect of it, and more
Hmm.
so just looking at how the brain learns and develops habits. And again, like my primary focus was on inhibitory systems. And so
Hmm.
I was looking in like, I was doing fibrophotometry recordings during backwards conditioning. So Pavlovian conditioning, but rather than it being tone, then reward, it's reward and then tone. And
Ah,
so essentially
interesting.
the tone becomes a signal that like reward is not there. And so it becomes an inhibitory signal. And so I was like recording activity during that time. And so then, so then now my goal is to sort of merge what I learned at UCLA about just like basic learning theory and, habit formation, decision making, all of that, and then combine it with what I knew at Iowa with drug addiction stuff. And then my added layer is adding the estrous cycle tracking. And partially because of our conversation, like you convinced me, you were like, it is relatively simple. And I was like, yeah, we can do that. And just from, there's the double reason of like, one, it hasn't been studied and it needs to be. But then also there's the benefit of like, it's kind of low hanging fruit then if we can do this. So that was sort of my reasoning behind it. And it's like an additional data point to collect with all the other things I'm collecting.
Yeah. So are you going to be doing just behavioral paradigms? Are you looking at any molecular markers or?
Just behavioral for the foreseeable future. I wouldn't rule out molecular markers. But yeah, so right now my process is I'm getting my behavior up and running with sucrose seeking sort of as my addictive like proxy, which there's more evidence that sugar has the addiction properties and all of that. My long-term goal is to get my DEA license and start to do drug stuff.
Cool. Obviously, which drugs are you trying to use?
I think practically opioids would be the best option for like science reasons. For the actual like having a drug on campus reasons, cocaine would be easier. So yeah, honestly, I haven't gotten that far into thinking about it. So I mean, I have experience with both cocaine and heroin in the research setting. So yeah, I think it would just kind of depend on like when it actually comes down to getting the DEA license, is it going to be a nightmare to get heroin? Because that would be prohibitive, but then also it would lead to the best, I mean, it's the most applicable research outcome, I would say, or like some opioid that's similar. So that's my goal, is to eventually add on the drugs. And then probably before that would be to add on some sort of brain manipulation. I would probably be doing like chemogenetics just because for cost purposes, that's going to be a little easier to sustain than optogenetics.
Because opto can be a little pricey.
I think the laser would be kind of prohibitive. And chemo would give me just like it'd be easiher to do with students too, and like all of those sorts of things.
And the students that you're working with, they're primarily undergraduate because of
all undergrad.
Very cool.
Which is it's awesome. I really like working with them, and plus they have like the excitement of research that sometimes we lose a little bit in grad school.
I feel like lose it, or does it get beat out of us?
Right. I know. It's like you kind of like become disillusioned with the whole thing, and they're all like, no, like this is so interesting, I want to contribute. I'm like, yes, that's I want to grab onto that and keep it.
Okay. And so with this, you're looking to bring in tracking the estrous cycle. And now are you going to be, I guess, looking at like all phases of the estrous cycle, or like what are you thinking for that?
So that's a good question. My initial thought, I have an experiment that I'm doing this summer where if I can feel confident and comfortable with the estrous tracking this spring, we would add it on to the experiment I'm already doing, just as a way to be like, okay, we're going to collect the data anyway, and then split the females at the end based on either whatever day that they were on the test day, or I could try to plan it so that they're all on the same phase. But a little bit of it kind of just depends on the practical nature. I guess long-term, it would be great to have like a map of like, we're doing this incubation of craving experiment, and we have a craving measurement for females during proestrus. We have a craving measurement for females during diestrus, metastrus, all of that. That would be like the ultimate goal. Realistically, I think it'd maybe be better this summer to test them all on the same day, just because I'm going to have limited numbers. And so in order to like see anything interesting, that'd be the best way. But I'm also open to any and all suggestions you have when it comes to that.
I'm so glad that you emailed me, because like I have more information about lavaging female rats than I think that I like wanted to have, just because I ended up doing it there was 2 projects where I was doing it, and I ended up doing it a lot longer than I actually needed to be doing it for the
Yeah.
first project. So then for my second project, which was completely circled around female hormones, it was like I knew exactly what I was doing. I knew what worked. I knew what didn't work. I
Right.
had like all the little like tips and tricks. And so I was able
Yes.
to get in there, do it really easily,
Mhm.
which was really nice. So I was
Yeah,
going through,
those tips and tricks would be so great
yeah,
to have.
yeah. So I was going through your email and then just kind of like bullet answering the questions that you asked. And then I will send all of this to you after, along with some
Perfect.
other pictures.
Mhm.
Um, so the first thing that I wanted to talk about is handling the animals. I know since like you've worked with animals before, you know that handling them before you start actually doing the procedure, so the lavage, is really important. How long do you typically handle your animals before?
Yeah. So for like not for like naive animals, new behavioral experiment,
Mhm.
the most I've done is usually like 3 days in a row and like
Okay.
usually like 5-ish minutes of like, picking them up, kind of poking them, moving them around a little
Hmm.
bit, getting them just used to that.
Yeah.
Um, from what I've seen, and it kind of sounds like you might get into this, it seems like that needs to be a little more involved
Hmm.
for this type of thing.
Yeah, that's
Um,
what I did. And I was probably a little more conservative with it,
mhm.
which I don't think is a bad thing because if the girls are stressed out, that's going to throw off their estrous cycle
Yeah.
to begin with. And I never had it happen to me, thank goodness. But I had some people tell me, like, oh, you got to be careful because, like, they'll skip phases, they'll stay stuck in one phase, and sometimes they'll go backwards. I never really saw that, but just like making sure that your girls are like as de-stressed as possible is going to be the best. Also, what light cycle are you going to have them on? Are they on like a normal light cycle, lights on during the day, or on a
Yeah.
reverse light cycle?
So right now they're on normal light
Hmm.
cycle. It's certainly I have the capabilities to obviously not like to change that. And I have two different rooms too. So like there's other
Oh,
rats
cool.
on normal
Yeah.
light cycle. So at least this spring while I'm figuring it out, I wouldn't be able to switch their light cycle. But when I
Hmm.
start the experiment in the summer, like if it would be better to have
Yeah.
reverse light cycle, we can do that.
Yeah, so I need to expose my bias. My like one of my projects was looking at diurnal circadian rhythms
Mhm.
and doing behavioral analysis. I think is best during their dark cycle, so during their active time, just to like play off of that.
Yeah.
They're nocturnal. And then in my readings, people had said because proestrus, when estrogen is high, proestrus is such a short window. It's only about 12 hours. The
Mhm.
literature says that the rats will most consistently hit that during their active time. So when the lights are off. And so if you're going to be doing your experiments during the day, you want to catch them in proestrus during like your day. It's going to be best to get them when the lights are off during their active time. In one of my projects, I had them on a normal light cycle and I was able to see proestrus during their like sleepy time, but it wasn't as consistent as proestrus happening during their active time.
Mhm.
Um,
Okay.
so that's definitely something to consider. And then, so going back to the handling, very important. I'm sure that you know this, but handling them at about the same time of day. And then when you're going to be doing the lavage, also doing that at the same time of day, as best you can. I mean, it's difficult. Like you're not going to get it right down to the hour, to the minute. And then for the handling, so what I would do is like with completely naive animals for the first like probably 2 or 3 days, I would just like put my hands in the cage and like let them smell me, get like used to me. And then I would start picking them up, really like holding them nice and close to me so that they could smell me, know that I'm not a threat. And then the after that, the position that you want to get the animals in to do, can you see this guy right here?
Yes. Yes, I can.
Okay. But so you want to hold the girls like belly up in the back of your hand and then putting them against your thigh, against your hip to kind of use that as like a counterbalance pressure to hold them.
Mhm.
I think is the best way to do it. But also like the more you do it, the more you will get a feel of it. And so in the initial handling, just like putting them in this position for like a couple days. And then what I started doing is with the pipettes. That was also something that you asked about. I
Mhm.
am definitely a proponent of using a pipette instead of doing a
Yeah.
smear. But instead of doing like this guy has some type of a pipette, I used a transfer pipette. So like one of
Oh,
these, like a 3 mil.
Okay,
Um,
perfect. That was what I was going to ask. Because I'm like,
mhm.
if I could just get a bunch of those,
Yeah,
that would be the
yeah,
most cost effective.
yeah, yeah.
Okay.
You want to make sure that you get one with a small gauge, because really don't need to get a lot of it. It's probably like a 3 ml should be good, and you only need like half a mil to a mil of, I use PBS. We'll talk about that in a little bit, but you want to get one with a small gauge. I remember when I first started doing this, we had like the big transfer pipettes, and I brought someone from a different lab with me. And they're like, whoa, why are you using that big of one?
Yeah.
And so like the smaller of a gauge you can get, the better, just because it'll, I mean, like it'll upset them less.
Yeah.
Um,
Okay.
and so then when you're handling them, start with just picking them up, getting them used to you, and then getting them in the position
Hmm.
first. And then what I would do is actually like get a pipette and I would place it on their vagina just to like
Hmm.
get them used to like, okay, you're going to be in the position and I'm going to be coming at you like this.
Yeah.
Um,
So like
and
without the actual penetrating, but just holding it there so they
yeah,
know.
yeah,
Yeah. Okay.
yeah. Just so that they could like get a feel for it, but also be like, it's okay, like I love you. I'm
Yeah.
not trying to
Be
hurt
safe.
you. Yeah. Yeah, so the transfer pipettes, and I would just keep like a big box of them in my animal room so that every time I went in, I could just go and do it. You also asked about solution to use. And so
Mhm.
I used 1X PBS, and this was really good. And I think that it is kind of important because PBS, it's isotonic. And so the cells, because you're going to be looking at the epithelial cells, the leukocytes, and if it's not isotonic, they're either going to swell or shrink. And that
Mhm.
can, if you're visually looking at the cells, that can be a little confusing if they're not just in in their natural state. And
Mhm.
so DI water, that's going to be hypotonic, that's going to make the cells swell, and that can either make you think that the girls are going to be in proestrus, or if they swell to the point where they're lice, then you might think that it's met-estrus.
Yeah.
So like PBS, just like a 1X PBS, we got a big box of 10X PBS, and then I would just dilute it and like keep a little Falcon tube with the pipettes in the animal room so that it's like it's there, it's ready to go. Just keep an eye on it when you would need to refill it. So you could use saline because it's also isotonic, but it's not buffered and it's not pH balanced the same
Mhm.
way that PBS is. The same way that we don't want our vagine pH to be off, like we don't want
Yeah.
that for the girls. And then also like there's a higher chance of inflammation happening with that versus PBS. So like I say PBS,
Okay.
that's what I use. That's what I think was really good. Another little hack is using these concave slides.
Mm.
They
Okay.
have little divots in it so that when you fill up the transfer pipette with like a mil, half a mil of PBS, and then you'll put it into the vagina, squirt it out, suck it back
Mhm.
up, squirt it out, suck it back up, maybe like 3 times, a couple of times.
Mhm.
And then you get all of the goo, all of the cells, all of the tissue, and then you just put like a little squirt onto your concave slide, and this just holds it really nicely. So the way that I did this is that I would kind of stage all of my slides because like you know what girls you have. So I would just put lab tape on the end of the slide and then I would number them with like corresponding with the girls. So
Yeah.
I would pull number 78 out and then I would say, okay, number 78 on the slide, take the sample, put it on the slide, put her back. And
Mhm.
so it's already laid out. And then you just clean the slides after each
Oh,
day.
and
So,
reuse them.
yeah,
Oh, okay. Okay.
yeah. So that was something that you were asking about, a preferred stain. I didn't use a stain. What type of microscope are you going to be using?
So I have just like a standard like Leica that goes up to
Mm,
100x. There are other microscopes that I have the ability to either use to see if it
mhm,
works and like even get like another um, objective if I need to.
mm.
If I could make it work on a
I
Leica with 100x, that
I
would be my preference.
I feel like 100X should work. I don't know if it was because of like the concave slide, but like I didn't need a stain. These are the images that I got from just
Okay.
on the day, putting it on the slide, putting it under the microscope. I had to play around with the microscope settings a little bit, just like the color, the contrast. Um, and then like you can see the cells really well. And what I would recommend doing, it made essentially a slide deck. And I would put the label of the animal. So this was like number 436. She was a wild type. And then every day that I was doing the lavage, I would have a picture. This kind of gets into, you were asking about the track by day method. And I guess that's kind of what I was doing. When you start, I think that it's good to just like consistently be lavaging the girls, one, so that they can get used to it, brings their cortisol levels down, is going to regulate their hormonal cycle. But then also like you're getting better at like actually doing the lavage, getting a good sample, and then being able to visually say like, oh, okay, that's proestrus, that's metaestrus, that's diestrus. And so I would take pictures every single day for all of the animals, and then I would pull that shot into this slide deck. And so each animal has their own slide. And so this was August 12th. And then this next day was August 17th. Like I might have missed a couple days. Or even if there were some days that I knew that I wasn't going to be recording, but like I would go and lavage the girls just so that they were regular, even if I wasn't taking a picture.
Yeah. Okay.
But then you'll be able to see it go from metaestrus into diestrus into proestrus.
To start to see the pattern a little bit like
Yeah,
that.
yeah,
Okay.
exactly. And so then it's really nice because when I was doing my experiments, it would be like, okay, well, I need to record from a proestrus girl. Number 436 is in diestrus today, but number 425, 434 is like she's in proestrus. So I'm going to record from her. And then this was also really nice because you'd be able to say like, oh, like I can see the cycling. And so I know that number 434 is going to be in diestrus on Tuesday. So
Yeah, that's a smart way to do it. I was definitely thinking one, maybe you have a suggestion for this too, about like a slide deck for examples
mm,
too.
mhm.
Especially since I'm going to be like training undergrads to help me
Yeah,
with this, like ones
yeah,
that are like this, like, are you looking for proestrus? This is what you're looking for, type
mhm,
thing. But I think
yeah.
visualizing each rat cycle too will be really helpful.
Yeah, yeah, and I can send you that image that
Yeah.
I already have it like in the email because those were like the best pictures of each phase that I got.
Okay.
And so when I was doing this, the two phases that I was looking at was proestrus and diestrus, because that's when estrogen is highest in proestrus and then estrogen is lowest in diestrus. And in a lot of ovarian hormone literature by Catherine Willie and Karen Frick, they have some really good, just like estrogen, it's a lot of physiology, but they kind of bookend the cycle with proestrus and diestrus. And so I would recommend doing that instead of trying to do like proestrus,
Each
diestrus,
day. Okay.
metaestrus,
Yeah.
just because there is a lot of overlap and because the rat estrus cycle is so short,
Short. Yeah.
being able to like definitively say like, yeah, she's in met estrus and going at proestrus, diestrus is really hitting at the estrogen question. But yeah, so that would be my recommendation.
Yeah.
But
Okay.
definitely look in the literature with people that are doing similar paradigms or looking at similar hypotheses. And also it's a little easier than
I
having
love
to,
that.
yeah,
If it's easier and also makes sense scientifically, that's the
yeah,
perfect combo.
yeah. So yeah, I'm trying to think if there's anything else. I'm just thinking about groups. Because I know for me, like I had my transgenic animals and my wild type animals, and then I needed diestrus, proestrus for each of those. How many animals are you going to be working with? How many groups? How many ends are you
Yeah,
thinking?
I think any given cohort would probably be like an N of either 8 or 16,
Okay.
depending on if I was doing females and males or if I just
Mm-hmm.
decided to do a whole female cohort. Uh,
Mm-hmm.
so yeah, probably at least 8, maybe
Yeah.
up to 16 for now.
Okay. Cool. Yeah. No, that's great.
Yeah,
That
how
would
many would you
be,
say for like a behavioral test? How many N to see like a good?
yeah. That's a good question. I guess I'm probably not the best person to answer that because all of my work was physiology.
Oh, yeah.
And like for each animal, I was getting like 100+ cells. And
Okay.
so my ends for that project, I had one, two, three, four. groups. I had like 40 animals in total. So I had,
Okay.
I think, 10 for each group.
But yeah, I can take a look at other people that do like the
Yeah.
throughput of behavior that I have to
Mhm.
get a similar idea.
Yeah, but I
Okay.
think anywhere between 10 and 16, like that sounds really good.
Yeah. Okay.
That would be super interesting if you're going to be looking at high estrogen, low estrogen compared to the males too.
Yeah.
Um,
And the first study we're doing is incubation of sucrose craving,
mhm,
because partially from anecdotal experience, but also the literature is just like, how does motivation for sugar change across the
mhm,
cycle? I
yeah,
would guess that it does.
mhm,
Um, so yeah.
yeah, super cool. That's going to be really interesting.
Yeah, I'm excited for it.
Hmm.
Though I mean, this has been super helpful because before we had this meeting, I was a little apprehensive, honestly, because
Hmm.
I'm like, oh my God, there's so much like I don't know, and I'm gonna have to
Hmm.
like still figure all of it out.
Yeah.
Which there's still a component of that, but
Hmm.
it's nice having these like the little tips and tricks. Yeah, because
Yeah.
looking it was like, well, you could use saline or DI water or PBS, and there was no reason as to which one would be better.
Yeah.
And it sounds like there's a really good reason as to why one would be better than the other,
Yeah.
but
Yeah.
it's not out there. So
Yeah, yeah. And also like throughout this whole process, like if you have more questions, feel free to get a hold of me and we can talk about it. But as like with anything in a lab setting, the more you do it, the more you will figure out what works and what doesn't. But it's pretty straightforward. When you start lavaging the ladies, like it's going to be uncomfy for them. It's also going to be uncomfy for anyone that's doing it, but
right.
they'll get used to it. You'll get used to it. I'm trying to
Mhm.
think another thing. Maybe it was just because I was a pansy, but I wore one of those. It wasn't like a chainmail glove, but
Oh,
you know, those like thicker gloves that you can wear.
mhm.
I wore one of those, and that was like my hand to hold the animal. So that was probably in my left hand. And then I had the transfer pipette in my right hand.
Just so
And
they don't like turn and try to,
yeah, I never had any close calls, but it just made me feel better.
yeah,
Um,
that'd be
and
it. Yeah, that'd probably be a good idea.
yeah. And then another little thing that I did is when I was doing it, like the order of operations, I would pull the animal out, validate, okay, this is number 78. I have number 78 slide. And then before picking up the animal, I would fill up the transfer pipette and just like set it down on
Mm,
the little table. The last thing that you want to do when you're holding an animal with one hand is be like futzing around trying to fill up
yeah,
the pipette. So
yeah, you want that like as short as possible.
yeah,
Yeah.
yeah.
And then
Um,
for imaging, did
mm,
you like about how much time between sample collection and collecting those images? Is that something you did immediately?
yes, I did
Yeah.
that immediately. I would walk from the animal facility, like I would go back downstairs to my lab. I would go right to the scope. You do want to do quick just because you don't want the samples to dry out. But there was sometimes like if someone would come up and be chit-chatting with me, maybe like my last two slides might dry out, you can rehydrate it with PBS.
Okay, that's good to know.
Yeah. But
And then
I wouldn't leave it for like a couple hours and
yeah, okay. Um, but that's good though. If we can get the image, then it doesn't necessarily like if we don't have time to actually analyze that, we can do
yeah,
that later, but at
yeah,
least we have the image.
yeah, yeah.
Yeah. Would you take like and maybe this changed as you got better at it, but like would you find that you took multiple pictures or would you try to just get like one good representative?
I would try to get one good representative. I would scan around and look for just like a large population of cells. But like for me, I would be lavaging 40 animals a day, every single day. And
Mm-hmm.
so I didn't want to
That kind of
have,
time. Yeah.
yeah, exactly. And so it's like, okay, like number 78, and you'll even see that like some days you just don't get any tissues, any cells, and like that is usually indicative of like a late met estrus going into
Okay.
diestrus. But yeah, that's also something that happens.
How
Yeah.
one of the things I've seen as like a risk for using the pipette method is the false pregnancies um from like stimulating the cervix on accident.
Ah,
Did you
interesting.
ever encounter that? And like, no? Okay.
I
They make it
didn't.
seem like it's like a huge common risk, but it sounds like maybe if you're just doing it wrong.
Yeah.
Um, okay. And then approximately like how many centimeters of the tip of the pipette would you say you insert?
Um, Sometimes, I mean, like the rats will let you know if it gets too deep, but I like it got in there.
Yeah,
And
okay.
especially since it's like a smaller one, it's not as upsetting to them.
Yeah,
But like you do want to make sure that it gets in there. I'm trying to think as far as like how many centimeters, maybe like 3 or 4.
okay.
Um,
That's helpful to know.
yeah,
I'm sure it's
yeah,
a little bit of like we'll get a feel for it
yeah,
too,
yeah.
but at least like a
Um,
good expectation before.
Yeah. And I guess something else with that is you want to make sure that you're not using too much PBS, like when you're kind of flushing out. Like you want to enough to pick up the cells, but you don't want it to be like so dilute where it's like a ton of PBS and then you're taking like a tiny little bit of it to
Mhm.
put onto the slide. It's really just like a good drop in, out, in, out, in, out, put it onto the slide. And like you'll see when the girls are in proestrus or estrus, like it's like really gooey. You can see the cells.
Okay.
You can just like you can see it. You want
Okay.
to
So
make
it's
sure
like
that
visual, even before the microscope, you have a sense for
yeah,
where they're at. Would you
yeah.
ever do visual inspection of like their genitalia? Because I know there's like suggestions that like you can sometimes classify by that alone.
I didn't.
No, okay.
Yeah,
I mean, I wouldn't ever use that alone.
yeah,
But if it was practical, like maybe keeping track of it, but that just kind of seems like it'd be too much. So,
yeah. I think that it would be like extra effort for not extra output.
right. Okay.
So,
Yeah. Okay. And I think if I have any other big questions, I mean, the answer is probably I do, even if I'm not thinking of one, but
mhm.
I can certainly, I know how to reach
Yeah,
you.
yeah. You just email me. And
Yeah.
then so yeah, I'll send you all of these little notes that I took. I'll also send you these images. This one image, I'm just like, I love it because I made it, but it's the different phases. And then I also have a little graphic of where estrogen and progesterone are in those different phases. So, and
Okay.
like for there's like more ovarian hormones than just estrogen and progesterone, but those are the two main ones. And then again,
Yeah.
like looking at proestrus and diestrus, like that's really dependent on estrogen. And for me, that was a big selling point in my write-ups and in my grants was that because estrogen is the primary ovarian hormone, there's so much literature on it. It has a really profound effect on the brain and the body that like that's why I was honing in on estrogen versus progesterone.
Progesterone. Yeah.
So
Okay.
yeah.
Okay. Um, and then you mentioned a couple names. If you think of any like really good papers,
Yeah.
um, I would love, if you can send those along, that's great. But don't,
Yeah.
don't dig too much if you don't have them.
No, I'll just direct you to Catherine Woolley, Karen Frick. I'll send you their stuff.
Okay.
They like wrote the book on estrogen. Catherine Woolley was a lot more electrophysiology, but Karen all over everything behavior. Those are just like foundational
Mhm.
foundational stuff.
Okay. Yeah, that's awesome.
Mhm.
Thank you so much. This
Yeah,
is
of
like,
course.
yeah, I really appreciate it.
All right, my beautiful brainy babes, that is all I have for you today. I have all of the important information that we talked about linked down in the show note. I also have a link to some hormone-free birth control. Natural Cycles is down there. Thank you all so much for listening. Stay smart, stay safe, and stay sexy. I love you, and I will see you so soon. Bye. Mwah.